Good morning, students. I'll be teaching genetics. Unit 1. Unit 1 includes historical development in the field of experimentation and their genetic significance. Then, cell cycle, in which we have to study mitosis, meiosis, control points in meiosis in life cycle of organisms. Then, Mendelian genetics, Mendel experiments design, monohybrid, dihybrid, trihybrid process, law of segregation, and principle of independent assortment. Verification of segregates by test and backcross, chromosomal theory of inheritance, then allelic interaction, concept of dominance, recessiveness, incomplete dominance, codominance, semi-dominance, pleiotropy, multiple alleles, pseudo-alleles, essential and lethal genes, penetrance, and expressivity. Level of genetics analysis. Genetics analysis can be categorized in three levels: classical genetics, molecular genetics, and population genetics. In classical genetics, it is genetics period prior to the discovery of structure of DNA. Molecular genetics start from the discovery of structure of DNA. And population genetics studied at the level of an entire population. In classical genetics, the period to the discovery of the structure of DNA is often spoken as the era of classical genetics. During this time, genetics pursued their science by analyzing the outcomes of crosses between different strains of organism, much as Mendel had done in his work with bees. In this type of analysis, genes are identified by studying the inheritance of trait differences, tall pea plant versus short pea plant. Okay. Now, the classical approach to the study of gene can also be coordinated with the studies of structure and the behavior of chromosomes, which are the cellular entities that contain gene. Then molecular genetics, which deals with the discovery of the structure of DNA. In this genetics, genetician entered into a new phase as the replication, expression, and mutations of gene could now be studied at the molecular level. This approach to genetic analysis was raised to a new level when it became possible to sequence DNA molecules easily. The gene's internal components, coding sequences, regulatory sequences, and non coding sequences can be identified, and the nature of the polypeptide encoded by the gene can be predicted. Okay, so this was the difference between classical and molecular genetics. Now, the population genetics. Genetics can also be studied at the level of entire population of an organism. Individuals within a population may carry different alleles of genes. Perhaps they carry different alleles of many genes. These differences make individuals genetically distinct, possibly even unique. Or we can say that the members of the population vary in their genetic makeup. Now, application of genetics to human endeavors. Genetics have various applications that are very useful for the human beings. Like it can be used in agriculture, in medicine, and it can be beneficial for the society. In agriculture, from the Mendel's era, it has been observed that there were various crosses done which were very beneficial for the farmers, and from that we approach to the genetic modified organisms and various other agricultural field. The selective breeding programs, now informed by the genetic theory, continue to play important roles in agriculture, like high yield variety of wheat, corn, rice, and many other plants have been developed by the breeders to feed a growing human population. And the selective breeding techniques have also been applied to the animals such as beef and dairy cattle, swine, sheep, and to the horticulture plants such as shade tree, turf grass, and garden flowers. By locating the genes for trait such as grains yield or disease resistance, breeder could now design a scheme to incorporate particular alleles into agricultural variety. For example, plants and animal breeders are now employing the techniques of molecular genetics to introduce genes from other species into crop plant and livestock. This process of changing the genetic makeup of organism was initially developed using test species such as fruit fly, but today it is widely used to augment the genetic material of many kinds of creatures. Plants and animals that have been altered by the introduction of foreign genes are called as genetic modified organisms. Nowadays, we are using various crops that were genetic modified. Cottons were modified, corns were modified, brinjals, tomatoes. These all are the examples of genetic modified plants. The best example is the BT corn. BT Here BT resembles the bacterium Bacillus thuringiensis, whose gene was introduced into the various crops to prevent them from the attack of various insect from the attack of insects. Now, role of genetics in medicine. In development of medicine, genetic play an important role, as without identifying the disease, the development of medicine is impossible. So identifying the faulty gene, mutant gene that causes the disease, then accordingly developing the medicine. The classical genetics has provided physician with a long list of disease that are caused by mutant gene. The study of these diseases began shortly after Mendel's work was rediscovered. That means when the Mendel's work was rediscovered, after that the scientists identified that there are various diseases that are caused by the mutant gene. For example,
Among the newborn, the disease known as phenylketonuria, a disorder of amino acid metabolism in which there is a formation of amino acid phenylalanine, and this disease occur only 1 in 10,000. And because of this disease, the untreated phenylketonuria can lead to the intellectual disability, mental disorder, behavior problem, and in some it may result in a much lighter skin and others. In newborn baby, it can show the small head, low birth weight, and others problem. Advances in molecular genetics are providing new ways of detecting mutant gene in individual. How it can be how the it can be identified? It can be identified by diagnostic test based on analysis of DNA, which are now readily available. For example, the hospitals or doctors can check our blood or swab for the presence of various mutant alleles, and accordingly, the medicine can be designed. Molecular genetics is also providing new ways to treat disease. For example, for decades, diabetic had to be given insulin obtained from the animal, usually pigs. But nowadays, it has been changed. Now, instead of obtaining insulin from the pig, it is now manufactured from the bacterial cell that carry the human insulin gene. Earlier, human growth hormone were isolated from cadavers, but now it is manufactured from bacterial cell, and this hormone is used to treat children who can't make sufficient amount of growth hormone. Many other medically important proteins are now routinely produced in bacterial cell that have been transformed with the appropriate human gene. Human gene therapy is another way in which molecular genetic technologies are used to treat disease. The, the strategy in this type of therapy is to insert a healthy, functional copy of particular gene into the cell of an individual who carries only mutant copies of that gene. And the inserted gene can then compensate for the faulty gene that the individual inherited. Nowadays, human gene therapy has mixed result. It can be used to treat cystic fibrosis disease, a respiratory disease in which lung cells have not been working properly. Now, the next application is role of genetics in society. As genetics play an important role in society in identifying individual identity, but the other role it can play is economic importance. Genetics provide an economic to the society. How it provides? As the discoveries from genetic research have initiated countless, countless business in the field of biotechnology industry. Companies that market pharmaceuticals and diagnostic tests or that provide services such as DNA profiling have contributed to the worldwide economic growth. Obviously, the much development in the genetic field, the much growth in the economy of the society. The next role of genetics in society is legal role. As the DNA sequences differ among individuals, and by analyzing these differences, people can be identified uniquely. Such analyses are now routinely used in many situations to test for paternity, to convict the guilty, and to identify the innocent or victims in the crime, that is in the paternity test, and identifying the criminals from the crime scene. All this can provide the legal role of genetics in the society. Next, the so this was the basic introduction about the genetics, where we have studied about the application of genetics, genetics levels, then organism that can be suited for the genetic analysis. In this introduction topic, the basic introduction part were included. Now, in the next lecture, we'll study about the cell division, cell cycle, their checkpoints. Okay, so you listen this audio properly and make your notes properly. I have discussed all the thing. Okay.